Chapter 1 Introduction.
Indonesia has a lot of potential to be developed. In Indonesia, there are 25,000 types of plants, which amount 10% of plants in the world. One of the plants that are abundant in Indonesia is frangipani plants from genus Plumeria. Today, many frangipani plants are attacked by a pest, so that these plants get sick or even die. As evidence, one of the research team has a frangipani plant that was infected by a pest, Paracoccus marginatus. According to Bioinsecticides, Paracoccus marginatus is a pest that attacks several horticultural crops in Bali. In its handling, the use of insecticides is an easy way. However, the resulting residue can enter the environmental component because the active ingredients are very difficult to decompose in nature. Therefore, new innovations are needed to eradicate mealybugs without having side effects for humans or the environmental, such as bio-insecticides. Based on the lab tests carried out at Udayana University FTP laboratory, the research team found that Canang waste contains metabolites such as alkaloids and flavonoids in quite a large amount. The research team chose Canang waste as bio-insecticides raw material because Canang waste is easy to obtain. Based on that background, the research team compiled a scientific paper entitled SAMSARA, Utilization of Canang waste extract as bio-insecticides in Eradicating White Pest, Paracoccus marginatus.
Chapter 2 Literature Reviews. Chapter 3 Research Methods.
Fifth, mortality test. Put the mealybug or Paracoccus marginatus into 5 jars that have been given airspace and frangipani leaf or Plumeria lentifolia. Each jar contains 10 mealybugs. Spray bio-insecticide from Canang waste into the 3 different jars with 3 different extra concentration and spray Paracoccus 500 EZ and aquades into the remaining jars. Give 30 minutes to get the ratio. Observe the number of dead or alive mealybugs in each jar for future analysis. 6. Drawing of conclusion. After the analysis process, a synthetic process is carried out by collecting and connecting the background, problem formulation, writing objective, and discussion carried out. Then a general conclusion is drawn and several things are recommended as an effort to transfer ideas.
Chapter 4 Research Results and Discussion. Chapter 5 Conclusion and Recommendation.
Let's start with the conclusion first. So these are the conclusions of our research. Based on the results of our research, has proven that Canang waste extract positively contains alkaloid compounds and 6,118.10 flavonoid compounds, which are able to eradicate white pest, Paracoccus marginatus. The Canang waste extract that we use has gone through several stages. The first one is Canang waste destruction stage. The second one is Canang waste dissolving with alcohol, and the last one is evaporation stage. 90% of Canang waste extract that has gone through the evaporation stage, dissolved with 10% of aquades, then the bioinsecticides is ready to use. The second one is based on the results of our mortality test, has proven that the combination of 90% of Canang waste extract with 10% of aquades is able to kill white pest, Paracoccus marginatus. This combination doesn't work as fast as pesticides. However, the application of Canang waste extract is able to kill white pest without causing any side effect to the environment. Next, recommendations. For the perfections of this research, we recommend some suggestions as follows.